Hi, I'm Salvador Cordova. I'm here with my friend Paul Price, and I'm going to give a presentation on a presentation on genetic entropy, the main principles simplified. So let me share my screen. So I have my slideshow. So the first principle of genetic entropy is that there are many more ways to break a machine than to make a machine. Now, a cell is an intricate chemical machine, according to a standard cell biology textbook by Bruce Alberts. And also, a tornado passing through a junkyard doesn't make machines of substantial sophistication like a 747 jetliner. 747 is an intricate mechanical, chemical, and electrical machine. So that's the first principle. There are many more ways to break a machine than to make a machine.
Do you know who first came up with that 747 metaphor there?
It was Fred Hoyle,
Fred
who's a
Hoyle.
physicist. Yep. And I'll
And he
actually
was
show a picture of him.
he's an atheist. So
Yes. Yes. Thank
just
you for
just
mentioning that. Um,
pantheist technically, actually, but still definitely not a Christian.
yes, definitely not. So that's important because even he was able to recognize this based on mathematics alone. Now, some people will complain that um, this thing about a tornado passing through a junkyard making a 747 jetliner, that natural selection will solve that problem. So this is principle number 2. Natural selection is falsely advertised to solve the problem of a tornado in a junkyard making a 747 jetliner. And speaking of Fred Hoyle, this is a quote, and it is cited as from Nature, prestigious scientific journal Nature, November 1981. He said, quote, "The chance that higher life forms might have emerged by chance is comparable with the chance that a tornado sweeping through a junkyard might assemble a Boeing 747 from the materials therein. I am at a loss to understand biologists' widespread compulsion to deny what seems to be obvious." And so this video will principally address uh, uh, principle number 2, that natural selection is falsely advertised to solve the problem of a tornado in a junkyard making a 747. This video illustrates how natural selection fails to overcome problems posed by systems requiring the simultaneous emergence of numerous well-matched parts, and therefore natural selection fails to be only marginally better than random mutation, and thus and this results in genetic entropy in species like humans. So I have to edit my slides. So thanks for the practice, Paul. Anyway, one way that I enjoy, that I think of this problem is Thomas Edison famously said, when he was trying to make a light bulb, he said, "I haven't failed. I've just found 10,000 ways that won't work." And so what that means is during the invention of Edison's light bulb, he found that there were 10,000 ways to slap parts together that won't work and only a few ways that will work to make a functional light bulb. Now, there may be an infinite number of ways to make light bulbs, but for every way to make a functional light bulb, there are a bazillion ways to put together random collections of atoms and not make a functioning light bulb at all. In comparable manner, even though there are an infinite number of ways to make a functioning airplane, it doesn't imply that there's a high probability that random chance will create a functioning airplane by something like a tornado passing through a junkyard. That may seem obvious, but it is probably instructive to think about the reasons why specifically. And the reason is that the aircraft parts need to have the right shape and size to fit together, and they need to be positioned and oriented in the right location for the aircraft to work. So that, that may be obvious, but I just wanted to state it. Things have to be the right size and shape and put in the right place. Random positioning and random actions just will not do that. And even at a more basic level, independent of whether this thing actually works, it's still unlikely that so many parts can connect so specifically together in an organized way. And that is why a tornado passing through a junkyard can't assemble a 747 jetliner or any other machine of even modest complexity. The problem of connecting airplane parts is analogous to connecting parts of a cell. And recall, a cell is an intricate chemical machine. This is a conceptual diagram from Bruce Albert's cell biology textbook. Two proteins that are parts of a cell. They're identified by the different shades of green. Note that for them to connect together, they need shapes that enable them to connect, plus the proteins need to have electrostatic charges on each of them in such a way that they pair together to enable parts to stick together. One can see that it is also highly desirable that proteins have specific shapes that allow them to partner with only certain other proteins and not others in order for the machinery of the cell to operate. And so this explains, one can kind of intuitively see then why protein probabilities are actually astronomically remote. There may be a bazillion ways to make proteins in theory, but to get them to connect together is actually not trivial. It's very hard for us to engineer this from scratch. So here is a picture of a life-critical molecular machine known as ATP synthase. We'd be dead without it. It's composed of several protein parts. Can you see my cursor here?
Yeah.
Composed of several protein parts that have to be properly shaped and connected in order for the machine to operate. Each of the major parts of this molecular machine are known as of ATP synthase are called proteins. So this, let me see, this beta here is a protein, this alpha, this thing labeled alpha is a protein. So is this other beta, it's another copy of it, but it's a separate part, but it's a copy of this one. And then you have these C proteins here, and there are multiple copies of it. And this is really interesting because the C proteins actually have to connect together and connect together in such a way that they form a perfectly sized ring that matches. I mean, that's just like, to me, that's amazing. Then there are these other parts here. So all these have to connect together. They have to be the right shape and the right size. I'm going to show another diagram. And this is also the ATP synthase, but it's in different colors and it's flipped upside down from the previous diagram. Yeah, I hope that didn't confuse you.
No, no.
All right, so I'm going to unshare my screen. Let me see. And what I'm going to do is I'm going to play a video. So first let me stop sharing my screen. And I'm going to play a video. It's only 2 minutes long. Let me cut my video.
A concentration gradient of protons across a membrane drives the synthesis of ATP by a large complex in the membrane called ATP synthase. ATP synthase, also called the F1-FO ATP synthase, has two main parts, F1, which protrudes into the cytoplasm, and FO, which sits in the membrane. FO consists of 12 identical subunits designated C. A central stalk, called the gamma subunit, extends from FO through F1. Surrounding the gamma subunit in F1 are 3 pairs of alpha and beta subunits, which service the sites of ATP synthesis. Other subunits designated A and B connect the F1 knob to the membrane. The proton gradient across the membrane drives FO to rotate. Protons enter through a channel in subunit A and move into the C subunits, causing FO to rotate in 120-degree steps around the axle. Protons are released from the C subunits into the cytoplasm. Note that as FO rotates, the axle also rotates, but F1 does not. F1 is held in place by its connection to the membrane through the A and B subunits. However, the axle turning inside F1 causes F1 to change conformation, catalyzing the formation of ATP from ADP and inorganic phosphate. An ATP molecule is produced for every 3 protons that cross the membrane through the ATP synthase channel. This function, in which a proton potential powers ATP production, is highly conserved. ATP synthase complexes with the same general structure can be found in the cell membranes of bacteria, in the intermembranes of mitochondria, and in the thylakoid membranes of chloroplasts.
I hope that was interesting, and I'll continue with my presentation. So that when one looks at the at all the parts and how they have to fit together, one can get the sense of the improbability of forming these shapes from random chance. So let me continue with the presentation. So proteins are made of strings of amino acids which spell the basic composition of the protein. This spelling is technically called the protein's primary structure. So that's the more technical term. I use spelling as more of a colloquial term. And you see the letters here in blue? That's actually the spelling of this part C, the protein here. So that's this is the literal spelling of this protein here. Now the spelling of the proteins determines the limits, and limits the possible shapes the protein can adopt. So just for completeness, the 3-dimensional aspect of the protein shape is described by using technical terms such as secondary, tertiary, and domain structures. What's amazing is that physics, the language of physics naturally translates the spelling of proteins to their range of shapes and folds. I use the word shape, but the technical term is fold. So when we have a different spelling, these proteins, many of them, will adopt a certain native fold naturally. Now I'm simplifying this a little bit for the sake of our discussion, but it'll suit our purposes. And if I actually gave more details, it would even be more amazing how these things work. So the idea is that the spelling governs the protein governs the shape. So there's another layer here. Genes are made of DNA, and they contain the recipe for spelling the proteins. So genes here on the left, you can see here, this is the gene for that protein I just showed that's spelled in blue here. So genes store the blueprint or recipe of proteins using an alphabet of only 4 letters. Now this language of DNA that only has an alphabet of 4 letters, Francis Collins called it the language of God, and he was the head of the Human Genome Project. But it turns out really that there are other languages in the cell. So many of them. DNA is only one of the languages. So we might even say that's only one of the languages of God. We have the language of protein with the alphabet of 20 letters of amino acids. So there has to be a translation process from DNA to get it to spell a protein. So now that we've quickly covered the spelling of proteins, both in the language of DNA and the language of proteins, we can now describe why natural selection fails as a theory. Instead of using the language of proteins and the alphabet of amino acids, I'll use, by way of analogy, the English vocabulary, which has certain ways of spelling each of the words in the vocabulary. The requirements of proteins being spelled such that they are the right shape and size in order to cooperate together. This corresponds to the idea of the correct spelling of words or strings within the accepted conventions and rules of human or computer languages. And in each of these languages, as a matter of principle, there can only be a few correctly spelled words relative to the infinite number of ways to misspell words. Hopefully that's straightforward so far. So I'm just going to use, so I'll use the spelling of the words in the English vocabulary for my example of genetic entropy, but one could use the spelling of any functional vocabulary and language to illustrate genetic entropy by way of analogy. So I hope you've eaten. There are different kinds of foods require different kinds of recipes. And these are two of my favorite foods, unfortunately. Tiramisu on the left and a McDonald's bacon double quarter pounder on the right. So I'm not gonna cover the entire recipe. I'll just use the ingredients list. So the ingredients list for tiramisu is eggs, mascarpone, espresso, ladyfingers, sugar, spiced rum, and cocoa powder. And the ingredients list for a McDonald's bacon double quarter pounder is 100% beef patty, a bun, pasteurized American cheese, applewood smoked bacon, ketchup, onions, and mustard. Now, do you think we could, through a process of randomly changing the letters of the words for the ingredients list in one recipe, we could make a readable, comprehensible ingredients list for another recipe? Let us call this transformation of one recipe to another macroevolution. So we're going to try to macroevolve the tiramisu to a McDonald's quarter pounder, or for that matter, any functional recipe, just a functional ingredients list. We'll do that by random changes to the letters. I'll call it a random mutation. I'll cover natural selection shortly. So, oh, by the way, this is somewhat analogous to that monkey on a typewriter with infinite time. And just as for fun or amusement, some scientists, University of Plymouth in 2003, tried to find out an answer to this question experimentally, whether a monkey could, given enough time, type out the works of Shakespeare. So they tried the experiment, and sure enough, the monkey banged away at the keyboard and typed nonsense. But even before the monkey got that far, he first tried to destroy the keyboard and did a lot of urinating on the keyboard. So that was kind of an aside.
Yeah.
Anyway, so let's go back to this tiramisu recipe, just the ingredients list. And those are the ingredients. And I'm just going to randomly change or randomly mutate the letters. And you can see I'm just changing the colors here, the letters that got mutated. And one can see where this is going. It's just going to be a disaster. We're not going to make anything viable. It just ends up as gibberish, much like what a monkey would type out. So the answer to our kind of scientific thought experiment is, can macroevolution of these words be accomplished through random mutation? And the answer is absolutely not. Absolutely not. And again, this corresponds to the recipes in the cell. in the DNA language and the protein language. And as people study this, and this is one publication from a poster that I helped put together, that's a diagram I put together, we can see the spelling of these of complex proteins, and we analyze it, and it's really hard to make something functional. Basically a protein that's spelled correctly so that it connects. So evolutionary biologists and many people on the internet will say, well, Sal, you're not including natural selection. That'll solve the problem. And this part will actually show why genetic entropy is a problem. Natural selection fails to do what people claim it does. So we'll go with Darwin here. Darwin is pictured on the left. In 1859, Darwin published an idea which claimed to circumvent the problem illustrated by my recipe example with a theory he called natural selection. At the time he formulated his theory, he really didn't understand proteins and genes in the way we do today. Herbert Spencer on the upper right framed Darwin's idea of natural selection with the phrase survival of the fittest. I'll use the phrase survival of the fittest a lot in this presentation. So let's approach again the question of whether natural selection will solve the problem of the tornado in a junkyard. Will apply survival of the fittest. So we take the original ingredients list, and I'll call it the parent, and make 3 copies of the parent. So far, so good. So now, each of the children will have a mutation the parents didn't have. And you could see each child has a different set of mutations. So now let's apply survival of the fittest. Well, it's kind of hard to say which one is the fittest, but all of them have problems. So let's just say hypothetically the fittest was child number 2. So child number 2 grows up, becomes a parent itself or herself. And the process repeats. And that parent has all these children, and the children have even more. So
and that was 2 out of 3 of your examples being selected away. So that's really heavy natural selection right there.
Well, the thing is, exactly, and it doesn't matter whether we're selecting them away or not. Do you see the basic problem? The problem is the fittest of the children is what survives. It's not the fittest genome. The fittest genome was back here in the first generation. So there's an equivocation going on. When they say survival of the fittest, it's really the survival of the children that are most fit.
It's the survival of the least mutated, but all of the options are still mutated.
Exactly. So what Spencer and Darwin left out was the idea is what happens to the parent where the parent has actually the best genome. Old age just gets to the parent, but the parent actually had the best genome. And so what seemed like an immutable idea, it's actually very flawed when we diagram it out like this. And the result is by the nth generation, you just have garbage. So natural selection doesn't work. Now there's a reason why it doesn't work. It's a little bit subtle. Going back to Edison, I haven't failed, just found 10,000 ways it won't work. Again, the principle here is that there are many more ways to break a machine than to make one. So this is Darwin's mistake. There are a bazillion ways to fail, only a few ways to succeed. If the opposite had been true, his theory would have actually worked, but it's not true. Therefore, Darwin's theory is false science, and that establishes the basic principles of genetic entropy. Now, there are more details to this that I've simplified. I used in my example, I used what's called a haploid model. When we use diploid models, we have to add a little bit more sophistication. And this is worked out by particularly by Bill Basner. Bill Basner is a mathematician, and he worked with famous geneticist John Sanford. They published some of their ideas in the Journal of Mathematical Biology. That particular article set records for that journal in terms of the number of downloads. People were just downloading it because it was so pioneering and landmark. They basically falsified almost 100 years of evolutionary theory. It was just based on false math, and it didn't account for the problems, such as I showed here. So that, in a nutshell, is kind of a simplified approach to genetic entropy and understanding probabilities of proteins. So if you have any comments or questions, I hope that was instructive. Thanks.
Yeah. Thank you. I did like it. And I think it does lay out the basics very well of, why on a fundamental level, the genome ultimately goes down, ultimately gets worse. So, yeah, thank you for that.
All right, well, I'm going to stop the recording, and thanks very much, Paul.